My name is Pradeep, and I am a bioimage analyst at the Center for Dynamic Imaging at the Walter and Eliza Hall Institute of Medical Research. And today I will be talking about my experiences using Dask for large image datasets. So microscopy is a very important tool in examining biological processes over various size and time scales. For example, we take 2D or 3D images, which are snapshots of these biological processes. Or we can take, if you want to look at dynamic phenomena, which most biological processes are, uh, we can take, say, 2D snapshots over time, or we can take 3D images, which preserves the spatial information over time. And what happens is, with the advances in microscopy, we can take um, images much faster with much higher resolution, which means there's an explosion of data where you go from megabyte to gigabyte range to now to terabytes, where our analytical pipelines we had and the way we looked at images do not scale well. So I will be talking about lattice light sheet microscopy. So at WeHi, there is a particular interest in, there's a big interest in studying malaria. And lattice light sheet microscopy is one such technique used for studying malaria. So lattice light sheet, what you need to know is that it's a technique that enables very high speed, high resolution imaging, and it has low phototoxicity, so it doesn't damage, because of the low amount of light needed, it doesn't damage the cells as much. So the idea is that we want to use lattice light sheet to study how malaria parasites, the mechanisms by which malaria parasites infect red blood cells. So you have the red blood cells in magenta and the malaria parasites in yellow. And the reason why they are yellow blobs are because those are actually the infected RBCs, or red blood cells. And what happens is the infected cells burst, the parasites are released, and then they infect the surrounding cells. And this release and invasion process is around 30 to 90 seconds. So, and it's not everywhere, like in the field of view. It's only in like certain subsets here you can see this invasion process. So not all of them actually burst at the same time. So we are interested in, when you're looking at this image, we are interested in extracting the subvolume so we can do the analysis. So this is a 2D max projection. So as an example of the release and invasion process, if you look at the white boxes, you can see them bursting right about here, and you can see the parasites being released, which then invades the surrounding cells. So another one is here and here as well. You can see bursting and invasion happening in real time. So an ideal workflow, we use the Zeiss microscope, so the file format is in CZI, that's the file. So ideal way would be we open this raw data. We need to do some processing. So in the context of lattice light sheet, because the images are acquired in a certain way, we need to, the images are distorted, so we need to remove those distortions and process it in a way we can then use it. So the ideal way would be to be able to interact with this data, select our regions of interest, extract and then save it for downstream analysis. So a big bottleneck is this process here because of a few factors. One, being a proprietary file format, CZI, we need to be dependent on the licensed software for processing, which can sometimes lead to delays because the limitation on number of licenses we can have and the number of people that can use it. And this processing can take a while. And because we need to process the whole volume, and ideally we only need that sub-volume or parts of the image. So the other aspect we're having is being able to manually interact with the data and see what happened. Like, did we get enough, say, events happening in that to then study our phenomenon of interest? Do we need to redo the experiment? Okay, can we, if we know what's happened, then we can plan our future experiments. So this increases the throughput once you start to get that feedback. And the other side is also that because we have these intermediary steps of processing the data, every time the data is processed, you're saving the intermediary processing step. So you're having this data duplication that happens. So you can imagine that if we have so many users and so many experiments happening, this can scale up quite very quickly, and you'll end up using a lot more space. So being able to release this from the start would be a very efficient way doing these analysis, the experiments and analysis. So this is where Dask comes in. So Dask is great for parallel processing of large image datasets. as I'm a beginner in this, I was really impressed by the number of blogs and tutorials, and the one of particular interest was the one where you could couple Napari with Dask and read the Dask arrays and display them in Napari in real time and be able to browse them on the go and load the data as we need. So the idea here is that, okay, based on the workflow I'm interested in, I could ideally be able to process the data on the fly, see what I need, and then annotate what I need and save the subvolumes of interest. So that's the workflow I've mentioned here. So what I would do is I'm planning to use this library, AISA's image.io. The reason I'm using this is you can read this proprietary file format into Dask array. So there's a command for that. So I can then create a 2D projection, use Napari to open my images, draw these regions of interest based on the events I'm interested in, extract the coordinates of these boxes, and then go back to the original volume and then extract those out. And then I can do downstream analysis in my software of choice when I need it. So this is an example of a data set where you have 139 frames in the time dimension, 155 slices, and the resolution is 2048 into 1985. So when I load this workflow into this image into my workflow, and when I perform the cropping based on the ROIs I've defined, for some reason it loads the whole data set into memory. So it's a 170 gig data set, so it ends up taking around 170 gigs of RAM. And this defeats the purpose of using Dask, so I wasn't sure if it was something to do with the way I was running this. So I tried rechunking into smaller sizes, and it still was consuming a lot of memory. So I looked it up on GitHub. I Googled it pretty much, and I came across the same issue on GitHub where someone used a different data set and had a similar issue. And the solution that was mentioned was if you run this command where you do a mapping and do a copy, an np.copy, what it does is it creates, from my understanding, it creates intermediary array of size which is limited to the size of the this chunk. And this led to significantly lower memory footprint, and this is what I was after. So I thought, okay, why not try different chunk sizes and use this command and see if I can make it the memory footprint even lower. So Dask has this really nice way of looking at CPU memory consumption and all. You can get these plots. So you have the CPU usage on the left, memory on the right y-axis, and time on the x-axis. And the memory usage is in the cyan trace. And when I use the default chunking from Dask, it used only around 30 to 40 gig of RAM, and I could do this in just over a minute. I could crop the subvolume. So I was interested in, okay, if that's the case, what if I actually reduce the chunk size and then do the cropping? So I reduced the chunk size, and and then I thought it would reduce the memory requirements, but if anything, it increased. Um, and I was like, okay, what if I try halving it across the different dimensions now? So I halved it across the Z dimension only. Memory was high, and it took over 2 minutes, close to 3. And when I halved it only across X and Y only, then I also took way more memory and time. So I'm not sure why this is the case. I'm not sure if it's because the differences in the dimension of the volume I crop and the chunk size contribute to this. But this is a bit surprising for me, and I'd love to know if anyone has any ideas of this. So there must be. I mean, I've been really enjoying using Dask. There's a lot of resources which have been great, but there must be a better way to do the reslicing. It could be something wrong with my code. I need to do a post issue on GitHub about this, and love to hear suggestions from the audience. The problem I have now is that all of this I was doing was on process data, which I was just testing it out. But now when I want to do the real-time processing, I realized the library I was using for reading the CZI images cannot read the raw data. There is some error coming up saying method not implemented, which I do not know why. So I've posted this issue on GitHub, and the developers said they're looking into it, but I've been seeing if I could solve this otherwise, and I've been delving into file formats and how to read the raw data from the file formats. So thank you for your attention. I'm very interested in any sort of comments or feedback.